Hello students, welcome back to the chromosomal basis of inheritance chapter. This is part 8a. So in this chapter we will learn about chromosomal mutation. We'll continue that. So before undergoing the before going through the chapter, I would want you to know understand that why this kind of mutations are happening in chromosome, like the altering in the numbers of chromosomes, and what is the reason behind the changes in number of chromosomes. You have to understand. For this, you have to recall the cell cycle, mitosis or meiosis, right? So because this numbers of change of chromosome is affecting the offspring, so in the organism the number of chromosome changes, so you have to recall this, specifically the meiosis 1 and meiosis 2, right? And if you recall, you will see that during metaphase the chromosomes are arranged in the equatorial plate, and as anaphase proceeds they get segregated. Okay, so at meiosis 1 the homologous chromosome segregated, and at meiosis 2 the sister chromatid gets segregated. If there is a change or if there is some defect in this segregation, it will affect the outcome of the numbers of chromosome, right? So that you have to recall. Okay, so as you know, the genome can also be modified on large scale by altering the number altering the chromosome number, or by the changing the chromosome or chromosome structure 1 or chromosome number 2. If you change the structure of chromosome, or if you change the number of chromosome, you can alter the whole genome, right? The whole genome will change. That's why. Okay, so the large scale variations are termed as chromosomal mutation or chromosomal aberration. So this can be, you can say that it's a chromosomal mutation, or you can say chromosomal aberration. Right, so what you have to understand, that if there is a change in chromosomal structure, that is, if, let me make it for you, if we have a chromosome and if suppose there is a deletion, this part is deleted for example, right? So the outcome will be that chromosome will be shorter, and those genes which are here will be deleted, because of which the outcome will be different. Second is, if the number of change of chromosome is changed, for example there may be addition or there may be deletion. For example, we have like a pair of chromosome, right? And if an additional chromosome adds, so now you have an extra number of chromosome, or it can be a deletion of, so you have single chromosome. So these kind of changes will bring the outcome, a different outcome, right? That will because of which the whole system will vary. Okay, so these gene mutations are changes that takes place within a gene, whereas chromosomal mutations are changes to a chromosomal region consist of many genes. That's important, what I told you. So if there is a change, if you delete a particular location, all the genes at this area are gone. All the genes for this particular area, or location is lost. So those genes which have some function will be lost, right? Okay, so it can be detected by microscopic examination, genetic analysis, or both. So we can do using microscope, we can see that there is a change in numbers. The numbers can be detected easily. You have to remember there is a process called karyotyping, right? Using karyotyping you can check that where is the number of chromosomes is varied. Is there an addition of chromosome, or is there a deletion of chromosome? You can check easily by doing the karyotyping. And genetic analysis will inform you or let you know whether the particular sequence of genes is available or it is lost, right? So in contrast, gene mutation are never detectable microscopically. You can never detect a gene mutation microscopically, because they are generally a point mutation or just a change in nucleotide, one nucleotide change. So it's very hard for you to detect. How can you see a nucleotide change in a microscope using a microscope, whereas the length of chromosomes or the number of chromosomes you can see using a microscope, right? So gene mutation is very hard to detect using microscope. However, chromosome mutation you can find using a microscope, and I already told you, just using karyotyping you can see The carrier you already know, right? Carrier is nucleus, right? Carrier stands for nucleus, so it's like studying the nucleus, right? So nucleus contains chromosomes, so you can see where is the number of chromosome changes or not. And if you remember amniocentesis, right? Using amniocentesis is used to understand the number of changes of chromosomes in the growing fetus. Okay, then further, chromosomal mutations are divided into two groups. Right, the two groups I told you already, right? So two groups. The number one will be the change in chromosome number. The first one is change in chromosome number, and second one will be the chromosome structure, right? So if there's a chromosome change in chromosome structure, that will consider as 2, and if the chromosome number change is considered as chromosome is known as change in numbers. Okay, so change numbers will be there. Okay, so let's start with the change in chromosome number. So as you know that each cell of a living organism possesses a fixed, important point, fixed number of chromosome. It varies in different species. That's important. For example, if we take humans, the human have 23 pairs of chromosomes. Right, but if you have a Drosophila, they have 4 pairs of chromosomes. So it's depending upon the species, the number of chromosomes varied. But however, within a species, generation to generation, the number of chromosomes stays same, and they stay same because of the two processes. The process number 1 is meiosis, and process number 2 is syngamy, right? So like we are diploid, so when we are diploid, there is a process called meiosis, right? And what meiosis do? They convert haploid cells, right? Sperm cells or ovum cells, right? They contain they make haploid cells. Right, and what happened in syngamy? In syngamy, these haploid cell from a male and a female fuses. They get fuses, and they restore the diploid condition, right? So these things happen over the period again and again. So try to maintaining the number of chromosome. Now the story of changing number of chromosome is because of meiosis. There's a defect in meiosis because of that. So if you remember disjunction, okay, just try to go through the chapter that they narrate on the cell division. So I suggest you to check for disjunction. Disjunction of chromosomes. So check for disjunction of chromosome, and you will understand that why the defect in disjunctions can cause us changes in number of chromosome, right? So okay, so come back. So even though some species of plants and animals are having identical numbers of chromosome, they will not be similar in character. Okay, so see, that's important, because what the sentence says is that even though like a plant, okay, like a plant A, let's consider this a plant and an animal, right? So like plant, if there's a plant which has like say is like 3 pairs of chromosomes, okay, 3 pairs of chromosomes they have, for example, and this animal also having 3 pairs of chromosomes. But these 3 pairs of plant and these 3 pairs of plant are not similar, right? They are not similar because the content of gene is totally different. And you already know that the chromosomes are condensed form of DNA, so the sequence will be totally different, so the outcome will be totally different. They are not in similar character. Okay, hence the number of chromosome will not differentiate the character of a species from one another, but the nature of hereditary material in chromosome that determines the character of the species. What the sentence trying to say is that the number of chromosome, if there's a change in number of chromosome, so it's not going to change the species from one to another, right? You will not, for example, a human is having one more chromosome. For example, some like we have the disease called so trisomic disease. So example 21st trisomy is there, a person is having one more copy of or one more number of 21st chromosome. So because the person is having one more number of chromosome number 21, so you cannot say that this is no more a human and he is something else. So that's not going to work, right? அப்படி ஆகாது இல்லையா? சரி, குரோமோசோம் நம்பர் வந்து ஒரு குரோமோசோம் அது அதிகமாக இருந்தாலோ இல்லை ஒரு குரோமோசோம் குறைவாக இருந்தாலோ அதனால வந்து நம்ம வந்து இது வேறு ஸ்பீஷீஸ்னு சொல்ல முடியாது, அதே ஸ்பீஷீஸ் தான். ரைட்டா? ஸோ குரோமோசோம் நம்பர் சேஞ்ச் ஆகிறதுனால ஒவ்வொரு ஸ்பீஷீஸ் வந்து இன்னொரு ஸ்பீஷீஸாக மாறாது. பட் குரோமோசோம் நம்பர் சேஞ்ச் ஆகிறதுனால உள்ளே இருக்க நியூக்ளியஸில் இருக்க ஹெரிடிட்டி மெட்டீரியலோட குவான்டிட்டி வந்து மாறும். அது மாறதுனால வந்து சேஞ்சஸ் வரும். ஓகே, ஸோ அடுத்து பாருங்கள், that sometimes the chromosome numbers of somatic cells are changed due to addition or elimination of individual chromosome or basic sets of chromosome. This condition is known as numerical chromosomal aberration or ploidy. சரியா? ஸோ somatic cellsல வந்து sometime chromosome number அதிகமாக இருக்கலாம், sometime chromosome number குறைவாக இருக்கலாம். ரைட்டா? ஸோ இது நம்ம என்ன சொல்லுவோம்னா இது நம்ம ploidyன்னு சொல்லுவோம். ரைட்டா? ஸோ ploidy வந்து ரெண்டாக பிரிச்சிருக்காங்க. சரியா? either Aneuploidy or Euploidy. சரியா? ஸோ what you have to remember that changes வந்து either additionனால நடக்கும். சரியா? ஸோ குரோமோசோம் நம்பர் எடிஷன் நடந்துச்சுன்னா குரோமோசோம் நம்பர் இன்க்ரீஸ் ஆகும், எலிமினேஷன் நடந்துச்சுன்னா குரோமோசோம் நம்பர் டிக்ரீஸ் ஆகும். ஃபார் எக்ஸாம்பிள், if I say that this is 2n+1 சொல்கிறேன்னா 2n நம்மளோட நார்மல் குரோமோசோம், தட் நம்ம குரோமோசோம் நம்பர் லைக் ஹியூமன்ஸ் பற்றி பேசினோன்னா 22n சொல்லும்போது we are talking about 46 numbers of chromosome, that is 23 pairs of chromosome. n addition 1 ஆட் ஆகிறதுனால மொத்தமாக 47 குரோமோசோம் இந்த கண்டிஷனில் இருக்கும். சரியா? இது வந்து அடிஷனால். இதே வந்து எலிமினேஷன் நடந்துச்சுன்னா லெட்ஸ் சே 2n-1 நடந்துச்சுன்னா ஸோ மொத்தமாக வந்து 46 குரோமோசோம்ஸ் இருக்கும். அண்ட் பிகாஸ் 1 குரோமோசோம் இஸ் லாஸ், ஸோ திஸ் வில் பி 45 குரோமோசோம்ஸ். சரியா? ஸோ ரெண்டு கண்டிஷனையும் பார்த்தாலுமே வந்து குரோமோசோம் நம்பர் வேரி ஆகுது. சரியா? இந்த மாதிரி வேரி ஆகிறது, குரோமோசோம் நம்பர் வேரி ஆகிறத வந்து நம்ம வந்து ப்ளாய்டின்னு சொல்லுவோம். சரியா? ஸோ ப்ளாய்டி 2 டைப்ஸ் இருக்குது. ஒன்று வந்து Aneuploidy and other one is Euploidy. Aneuploidyல என்னென்னா இண்டிவிஜுவல் குரோமோசோம் நம்பர் சேஞ்ச் ஆகும். சரியா? செட்டாக மாறாது, இண்டிவிஜுவலாக மாறும். நம்ம பார்ப்போம். சரியா? ஸோ இந்த பர்டிகுலர் டாப்பிக்கில் வந்து we are focusing mainly on the individual change வந்து நம்ம ஃபோக்கஸ் பண்ணுறோம். ரைட்டா? ஓகே, தட் இஸ் Aneuploidy நம்ம மெயின்லி ஃபோக்கஸ் பண்ணுறோம். ஓகே, ஸோ லெட்ஸ் see next. ஓகே, நம்ம ப்ளாய்டி நம்ம பேசும்போது சரியா